Welcome back, Biomonsters. Today we're going to be focusing on monohybrid crosses. I hope that you remember from our discussion of Gregor Mendel and his work with peas this term. We've talked about it before. So let's go ahead and quickly review what a monohybrid cross is. Remember the prefix mono stands for the number 1. In a monohybrid cross, we're focusing on one trait. So as we're doing crosses and looking at sexual reproduction and trying to see offspring from one generation to the next, we're focusing on one thing. The example that we talked about in our last video, the one trait that we focused on in the monohybrid crosses that Mendel was focusing on, was the height of pea plants. So we're going to continue to talk about monohybrid crosses today, but today we're going to be focusing on specific tools to help us better predict what the outcomes might be when we cross two parents with known genotypes. So let's do a quick review before we actually start talking about this new tool to make sure that we understand all the vocabulary. The key here is that if you don't understand the vocab, you won't be able to address any of the genetics word problems that you're going to see today and in the future. So let's look at the first question. It says, what is the term used to describe an organism's physical appearance? Now we've seen this word physical appearance before, and when we did, we told you to pay attention to the PH in the word physical. Physical sounds like the word phenotype. So something that you can see, if you can see the organism's physical appearance, it's the phenotype. So let's go ahead and say that you can see this so we don't get confused. The next question asks, what is the term used to describe genes that an organism has for a trait? And again, gene sounds like the term or the vocabulary term that represents the genes inside of an organism, and those genes inside of an organism are called a genotype. So genotype sounds like the word gene, phenotype sounds like the word physical. The next question, what is the term used to describe when an organism has two of the same alleles for a trait? Remember, this term allele means a gene, and that gene is always represented by a specific type of letter. And remember, those letters can be capital or they can be lowercase. But in this case, we're simply asking, what is it called when both of the letters are the same? So when they look like big T, or they could also look like little t. When they're the same, we collectively refer to this as homozygous. Remember, the prefix homo means same. Now our last review question asks, what is the term used to describe when an organism has two different alleles for a trait? So in this case, there's only one option. You have to be big T, little t. If you're big T, little t, the term that we're going to use to describe that is heterozygous. And if you remember from our last discussion, that prefix hetero means other, or we could also say that it means different. So we have one capital letter and one lowercase letter. If you get confused at all today when we start talking about our word problems, I want you to flip back to this section of your notes and review this key vocabulary. And so what I want you to do on your Cornell notes is to go ahead and highlight this and say vocabulary for genetics word problems, so you know where to go if you get stuck. During our Intro to Genetics video, we talked about the concept of reproduction being a lot like playing the lottery. The reason why we say reproduction, specifically sexual reproduction, is a lot like playing the lottery is because something called crossing over that takes place during meiosis when we're actually forming our gametes makes predicting the outcome of fertilization imperfect. Because remember, crossing over creates genetic variation. That means that we are intentionally mixing up our DNA to get new combinations of genes. Next, another reason why we say that sexual reproduction is a lot like playing the lottery is because of random selection. This basically means that we never control which sperm fertilizes which egg. That happens randomly. So it makes predicting the outcome of fertilization imperfect because every egg that a female produces is genetically unique because of crossing over, and every sperm that a male produces is genetically unique because of crossing over. So we're going to take crossing over add random selection to the mix, which makes it even more difficult to predict what you're exactly going to get every single time a fertilization event happens. Lastly, predicting the outcome of the offspring can be done when the genotype of both parents are known. So we can increase the likelihood of figuring out what we are actually going to get when we know what the genes or the alleles or the genotypes actually are of both parents before we actually do our cross. So what we're going to be focusing on today is knowing that information, so being given the genotype of the parents and figuring out what the probability is of getting specific type of children if a fertilization event were to actually happen. So let's talk about the tool that we're going to use. We're going to be using something called Punnett squares, and you guys have probably heard of this and used them in middle school. Well, our job for you is to become Punnett square experts, to be able to predict what a cross between two parents might lead to in the next generation. So Punnett squares are used to show what the offspring from two parents could look like and what genes they could have for a trait. It's really important that we use the word could. The reason why we're focusing on the word could is because Punnett squares do not tell us what we're actually going to get. Instead, Punnett squares have the ability to show us possible combinations. So it basically tells us what the probability is that each event will happen. And probability is pretty simple because we're only focusing with a handful of numbers. So our probability will either be 0% that we will get a specific genotype or phenotype, 25%, 50%, 75%, or lastly, 100%. So the thing is, even when we use our Punnett squares and we use them correctly, we're never going to know exactly what we're going to get. And the reason why we're never going to know exactly what we're going to get is because genetics and a fertilization event is like flipping a coin. We don't always know the exact ratio, so we're just predicting what the potential outcomes could be. All right, so here's our monohybrid cross example. So if you look at our example, it says a tall allele is dominant to a short allele in pea plants. So we're going to cross two heterozygous tall pea plants, and here's our picture down here. We have our female and we have our male, obviously because this is sexual reproduction. Our job today is to figure out how to unlock a word problem like this and know where to start so that we can begin the process of figuring out how to populate our Punnett square and then predict possible outcomes for specific genotypes and phenotypes. But the good news is that we have some specific steps that we want you to follow. So whenever you're solving monohybrid crosses, the first thing that we want you to do is hammer out the problem. We want you to do this every single time you see a word problem. And when we say hammer out the problem, basically we're talking about reading as well as test strategies. So we want you to do things like underline. We want you to circle. And if you have a highlighter, we want you to highlight the important information as you're reading through the word problem, because the word problems could get pretty lengthy and it's easy to get confused. The next thing that we want you to do is make a key. A key is really important here because the key will prevent confusion. So let's go ahead and write that. It's going to prevent confusion. If you think about keys that are found on graphs or that are found on maps, keys show you what things stand for. And so we want you to make a key to tell us what letters stand for what parents, what combinations of letters we might have, so that as we're starting to populate our Punnett square again, we don't get confused. The next thing that we want you to do is find your parents. And here's the key. People sometimes get confused. If we're talking about monohybrid crosses, we're talking about sexual reproduction. So if we're doing sexual reproduction, we're always talking about two parents. So when we tell you to find your parents, you're always looking for two. So if you can't find two, you know that you've made a mistake. If you find more than two, you know that there's another mistake there too. So in order to find your parents, you have to use your reading strategies, figure out what letters you're going to use, then you're going to find your parents and write down their genotypes. You're going to ask yourself, are they homozygous or are they heterozygous? And let's quickly go over the difference between these two so we don't get confused. Homozygous, again, means same. So for homozygous, you're going to have either 2 capital letters or 2 lowercase letters. If it's heterozygous, hetero means other, or we could also say that it means different. So you're going to be looking for a capital and a lowercase combination. The next thing that you're going to do is fill in your Punnett square. And remember, your Punnett square is simply the structure that looks like this. Your parents are going to go on the outside and your possible children are going to go on the inside. The last thing that you're going to do is answer any of the questions that might be associated with the word problem. And we're going to practice all of this stuff right now. So the first thing that we're going to do is we're going to look at the word problem that we were just given and we're going to hammer it out. So I'm going to go through using my reading strategies the way that we've always done in class to figure out what the important terms are, who the parents are, and all of that other pertinent information. So as I'm reading, it says the tall allele is dominant. That's important. So anytime something is tall, I know that it's going to be dominant. That's important information. Which means that anytime something is short, it's going to have to be recessive. So I'm going to go ahead and write that down so that I don't get confused. Okay? The next thing that I'm going to do is I'm going to remind myself if something is dominant, is it going to be a capital or a lowercase? Well, I know if something's dominant, it's always going to be a capital letter. So I'm going to write that down so I don't get confused. And if something's recessive, I know that that's always going to be a lowercase letter. Then I'm going to read on. It says cross 2 heterozygous tall pea plants. So both of my parents are going to be heterozygous. So now I need to ask myself, if something is heterozygous, what does it look like? Well, I know this prefix hetero means other, or it means different. So that means I'm going to have 1 capital letter, and I'm also going to have 1 lowercase letter. All right, so I picked out all the important parts of my question and have done step 1. Now here's your first stop and jot. Go ahead and pause the video, answer the questions on your day sheet, and when you're done, call your instructor over to check for correctness. All right, now that we've hammered out our problem, the next thing that we want to do is make our key. In order to make our key, we have to figure out what letters we want to use and what letters stand for dominant traits and what letters stand for a recessive form of the trait. So here it says tall is dominant to short and pea plants. We figured this out by hammering out the question in step number 1. Because tall is the dominant allele, we're going to use the capital letter. And for our case, we're just going to go ahead and use T for tall, capital T. So I'm going to put capital T equals tall, and I'm going to write that this is dominant, and you should be writing the same thing in your notes, which means that the lowercase is going to stand for short, and that is going to be recessive. And again, just so that we don't get confused, this is the key that needs to be included next to all of your word problems. So I'm going to go ahead and highlight that. I want you to do the same thing on your paper and when you're working with your own word problems. All right, step 3. For step 3, we want to find our parent. So let's go ahead and look at our word problem again. So we have tall is dominant to short and pea plants, and we're going to cross 2 heterozygous tall pea plants. So that I know my parents are my 2 heterozygous genotypes. So what I need to do is figure out what that heterozygous genotype is going to look like. And I know heterozygous means different. So that means I'm going to have 1 capital letter and 1 lowercase letter. So let's go ahead and see what our female plant's genotype is going to be. So here we go. We have a big T, little t. So this is my genotype. It's my genes. And then for my male, I'm going to have a big T and also have a little t. And again, just so we don't get confused, this is still our genotype. Here's your next stop and jot. Go ahead and pause the video, and when you think you have the right answer to the question on your day sheet, call your instructor over to check it for correctness. All right, for step 4, all you're going to do is draw in your Punnett square. And just as a preview for your Punnett square, the only thing that are ever going to go on the inside of the Punnett square are your possible offspring. So this is only going to be offspring found inside these boxes. And over here, you're going to have parent 1. And on top, you're going to have parent number 2. What I'm going to do once I draw my Punnett square is set it up. So now I need to be thinking about my key as well as my parents that I identified in my previous steps. So I'm going to focus on the female first. You don't have to. That's just the order that I'm going to go in. And now if I remember correctly, my female genotype happens to be big T little t because it's heterozygous. So now that I know what my ovum combination is, I'm going to go ahead and put it along the side of my Punnett square. Now again, you guys could choose to put it here instead if you wanted to, but we're just going to go ahead and put it here for now. Now what I'm going to do is focus on my next parent, which happens to be the male. And in this particular case, the male and the female have the same exact genotype. They're both heterozygous, so big T little t. So now that I have my sperm combination, I'm going to go ahead and put them on top of my Punnett square. Now that I have my allele combinations from my sperm and from my egg on the outside of my Punnett square, I'm ready to go ahead and do my cross. All right, now that I have my parents set up on the outside, I'm going to go ahead and do my cross. So be sure to pay attention to where things are moving and how I'm moving them. So for my first cross, what I'm going to do is I'm going to ask myself what's on the outside and on top of every single one of these individual boxes. So I'm going to focus on box number 1 first within my Punnett square. I have a capital T over here on the outside. I'm going to go ahead and push that inside into that first box. So here comes my first letter that needs to go in my first Punnett square. Then I'm going to look at my next cross. So the next thing I'm going to do is look at what alleles on the top of the box, and I have another capital T. So I'm going to go ahead and bring that into my Punnett square as well. So my first Punnett square box is already taken care of. I have big T, big T. So there my first cross is done. Now I have 3 more to do, but I'm going to follow along in the same fashion. So for my second cross, I'm going to be looking at this box. Again, I'm going to ask myself, what's on the left-hand side and what's on the top? On the left-hand side, I have a capital T, so I'm going to go ahead and bring that into the box. And then on the top, I have a lowercase t. I'm going to go ahead and put that inside of my box as well. Now I have my allele or my genotype combination in my second box, so my second cross is done. I'm going to do the same thing now for my third box. So for my third cross, again, I'm going to ask myself what's on the left and what's on the top. One thing that you want to pay attention to is that you still want to follow putting things in the same order that you would find them if you were writing a sentence. Remember, the very first letter of every sentence needs to be capitalized. The same thing is true inside of your boxes. So if you have a heterozygous combination, a big T, little t, you always want to make sure that your big T goes first. So we're going to go ahead and bring in our big T that's found at the top. So we're going in the opposite order that we went through before because we want to put our capital letter first. Then we're going to ask ourselves what's on the side. We have a lowercase t, and here it is coming into our Punnett square. So now this genotype combination is done. We have one more cross to do. In our fourth and last cross, again, we're going to ask ourselves, what's on the side of this box and what's on the top, and we're going to bring those two things in. So I'm going to bring in my lowercase t inside my Punnett square in my fourth cross, and I also have a capital T on the top, so I'm going to bring that in as well. So my last box is going to have a lowercase t, lowercase t. All right, here's your next stop and jot. Go ahead and pause the video, answer the questions on your day sheet, and when you think you have the right answer, call your instructor over to check for correctness. For step 5, what you need to do now is look at the Punnett square that you've created and answer the questions that might be asked of you on your word problem. So here are some examples of questions that you could be asked, and I want you to go ahead and see if you can answer these questions along with me. So one question that you might be asked is, what are my genotypes? Now remember, genotypes are your genes, and so these are your letter combinations. I'm going to go ahead and write that. Your letter combinations. So what are my possible genotypes? All I'm going to do is look inside of my Punnett square. So one possible combination is big T, big T. So I'm going to write that down. If I go to my next box, I notice there's another option, which is big T, little t. So it's possible to get a big T, little t gene combination. Same thing is true here, so these are the same. Now I'm going to look at my last box. It's also possible with these two parents to get offspring that are little t, little t. The next question is, what is my genotypic ratio? Well, to determine my genotypic ratio, all I have to do is count it up. So I'm going to look at my first box. How many boxes look like big T, big T? If I go through, I see it's only one. So I'm going to write 1. Then I'm going to look at my next box, big T, little t. How many boxes look exactly like this? Well, I have this one and I have this one. So that's 2. And then lastly, I'm going to look at my last box. How many boxes have a little t, and I notice that it's only 1. So my genotypic ratio is going to be 1 to 2 to 1. My next question asks, what are the phenotypes of my offspring? Well, phenotype, if you remember, means physical. So it's what it looks like, so what you see. The easiest way to do this is to go through and just go ahead and write down what each one of these genotypic combinations will actually give you. Now remember, big T equals tall, it's dominant, and little t equals short for our key. So if I'm big T, I know that this is going to be tall. Big T, little t, remember this big guy is stronger than this little guy, so it masks it, so you're only going to see the capital T, so this is also going to be tall. Because these two are exactly the same, I know this one is also going to be tall. And then lastly, I have little t, So the only way to see the recessive allele or the recessive trait is to have 2 lowercase letters. So I do have a box where it's possible in my Punnett square for these two parents to make a baby that is short. So what are my possible phenotypes? It's possible for them to produce tall offspring, but it's also possible for them to produce short offspring. The next question asks, what is my phenotypic ratio? And to determine my ratio, I'm going to do the same thing again. How many boxes represent babies that are tall? So I'm just going to count 1, 2, 3. So 3 of my babies are going to be tall, and 1 of my babies is going to be short. So that is my phenotypic ratio. The last question asks, how many are homozygous and how many are heterozygous? Now, homozygous again means same. So how many are capital or lowercase? So I'm going to go through. I have 1, 2. So it's possible to have 2 homozygous individuals in this Punnett square. How many are heterozygous? Remember, hetero means other or different. So if I look closely, it's possible to have 2 heterozygous individuals within my Punnett square. One question that this isn't asking about is the probability or the percentage. So let's go ahead and fill that in. If I asked you what the genotypic probability, you would tell me that there is a 25% chance of getting big T. There is a 50% chance of getting big T, because I have 2 boxes, and again a 25% chance of getting little t. So if you treat your Punnett square like $1, 25 cents, 50 cents, 75 cents, $1, it's really easy to figure out the ratios. What if I asked you the ratio, or I'm sorry, the probability, the phenotype probability? So what is the probability of being tall? The probability of being tall, I have 1, 2, 3 boxes, so that's 25, 50, 75. So there's a 75% chance that these 2 parents will have a baby that is tall. And if I look, there's only a 25% chance of these parents having a short baby. Now, it's possible for them to have either, but there's a greater probability that the babies will be tall. We've gotten to our next stop and jot. Go ahead and pause the video, see if you can answer the questions on your paper, and when you're done, call your instructor over to check for correctness. All right, here's another example. It's your turn. Think about the steps that we've talked about, and let's go ahead and see if we can answer this question. So it says, in guinea pigs, brown fur is dominant to white fur. Cross a heterozygous brown guinea pig with a white guinea pig. So the first thing that we want to do is hammer. just so there's no confusion. So I'm going to go through and read it with reading strategies. So brown is dominant. So I know that we're going to use a capital letter because dominant is capital. To white fur, so that means white is automatically going to be recessive. And if it's recessive, we know we're going to use a lowercase letter. Then I'm going to read on, it says cross a heterozygous. Heterozygous means different. Brown guinea pig with a white guinea pig, okay? So we have our two parents. This is going to be parent 1, and this is parent number 2. The next thing that we need to do is figure out our key. And so here we go. We have to pick some letters. I'm going to go ahead for brown because it's our dominant color. I'm going to say we're going to go ahead and use B. And for white, our capital B, we're going to use a lowercase. So again, this is dominant because it's capital. And this is lowercase because it's recessive. After we do that, the next thing that we need to do is figure out who our parents are. So let's go ahead and look at parent number 1. Parent number 1 says it's heterozygous. So we know that our letters have to be one capital and one lowercase. So here comes parent number 1. I'm going to go ahead and label that so I don't get confused. Parent number 1. Now I need to figure out what parent number 2 is. Well, I know parent number 2 is white, but this tells me white is recessive. There's only one way to see a recessive phenotype, remember, it's to have 2 lowercase letters, because anytime you have a capital with a lowercase, the lowercase disappears. So that means that our two parents are now going to be big B, little b, so heterozygous, and little b, which is parent number 2, and this is going to be homozygous because they're the same, but they're lowercase, so we're going to say that this is a homozygous recessive genotype. The next thing that we need to do is put it into a Punnett square. So here we go. We have our parents already listed. They're already here at the top, parent number 1 and parent number 2. And again, remember, it doesn't matter which parent you put where. I'm going to go ahead and put my first parent up here at the top, big B, little b, and my second parent over here on the left-hand side. Now, once I've done that, the next thing that I have to do is fill in and do my 4 crosses. Remember, whatever's on the side goes in, and whatever's on top goes in, but we want to put our capital letter first. So in this first box, I'm going to bring down my big B and bring over my little b. I'm going to do the same thing for my next quadrant. So I'm going to go ahead and bring over the little b, bring down the little b. I'm going to go ahead and do it again for my next quadrant, bring in my alleles that are on the top and the bottom. Again, I'm going to put my capital letter first, my lowercase letter second, and then I have one more cross to do, and in my last cross, I'm going to do the exact same thing. Bring in whatever's on the side, little b, and whatever's on top, little b. Once I've done that, what I like to do is go back and start to fill in information. So I know if I'm big B, little b, remember big B is dominant, it stands for brown from our key. Little b is recessive, it stands for white. So let's go ahead and fill in what each one of these babies is going to look like. So if I'm big B, little b, I know this one has to be brown because this big B is more powerful, so it's going to go ahead and mask this one. For my next one, little b, I know that this is going to be white. And then these 2 boxes look exactly like this, so I'm going to go ahead and fill them in as well. Now that we've filled in the Punnett square together, go ahead and do your last stop and jot, answer the questions on your day sheet, and when you think you have the right answer, call your instructor over to check for correctness. Thanks guys, and good luck.